Hello, Marco. I think I see your pinky there.
Yes, sir, that's me.
Seems everyone understood us to say there was no lecture today or Friday.
There's somebody downtown, but nobody here yet.
Okay. I'm going to go ahead and get started anyway. We can get it recorded and posted, and then I'll work on the other recording for posting tomorrow.
Okay, sounds good.
And everything should be in order. Have you had, and I still haven't had a chance to correspond with Nayeli on getting the exam questions typed in. Have you had a chance to do that?
No, sir, I haven't. I understood that you wanted me to wait for her to do it.
Okay.
I'd be happy to.
Well, if you have a chance to work on a couple of them, then I'll ask her to interface with you.
Okay, that sounds good.
I'll send her an email directly right after class.
Okay.
Okay, let's get started. You seeing me or the desktop there? And I no longer have your voice. Okay, I'm going to reboot.
I've got it down.
Okay. Thanks. So we begin lecture 25, urinary genital system 2, the primary sex organs. And we begin by thinking about the earliest state where there is an indifferent feature of the developing embryo, and we say embryo because we're still in week 5. This indifferent stage is one where the cytogenesis and histogenesis and organogenesis has not reached a point because the genotype is not yet expressed for us to differentiate male from female. It doesn't mean the genotype doesn't exist, but I was saying it hasn't been expressed as a phenotype yet. So in week 5, there are ridges that appear, and we saw this introduced in a previous slide in discussing the urinary system. So it's on the medial side, actually the anterior or ventral medial side that we had pointed out of the mesonephros with the primary sex cords that contain the germ cells. We'll see this figure again, but we're trying to picture it on that previous slide that we had looked at when studying the development of the urinary system. And it has extensions, and these are projections into the ridge. So we know that there's going to be some association between urinary and reproductive systems, so there's no shock that these are developing relatively near each other. But we know that a lot of things are going to happen because we produce 2 different gonads, and based upon the genotype and under normal conditions, of course, and that those sets of gonads end up in slightly different locations. They're not way up in the abdominal cavity. They're not just below the kidneys either. Even the ovaries fall lower in the abdominal cavity at the edge of the pelvic cavity, the pelvic brim as it's called. But in males, we know there will be a descent into the scrotum. So the indifferent gonads develop a cortex and a medulla. So there is a differentiation on surface to deep. And there will be an ultimate differentiation at this emerging from this still indifferent stage, but different parts will contribute to different things. So when the genotype is XX, the cortex of this gonadal ridge, if we can call it that, comfortably will become the ovary. The medulla will develop into the testes and a genotypic male. The primordium is just getting into that area where it needs to be and then is waiting for the signal. And there's clustering of cells, and there will be cells coming from different origins, different locations, and germ cell layers to contribute overall, and we'll see this eventually as we continue through this discussion. So in week 4, the primordial germ cells migrate from the yolk sac. So this is endodermal origin from the yolk sac, and we'll follow that migration into the abdominal cavity for location on the mesonephros to contribute to the formation of the gonadal ridge. So the sex becomes determined by the productions of a testes determining factor, TDF, in different gonads. And this, of course, will occur only when the Y axis, the Y chromosome is present. So the testes determining factor will cause primary sex cords to develop along the lines of the male lineage, in other words, to become testes. So we had stated before that we will see that the default development is along the lines of female differentiation lineage. And if the Y chromosome is present, then there will be factors contributing to the development along the lines of the male. So here we see this route of the primordial germ cells. This is just showing the complete trail. I don't think we end up with this long length of cells that some are already here and some are still moving in. I think there's a little bit of a clustered migration, but this shows the entire route from the endodermal sac all the way into the, from the yolk sac all the way into the intra-abdominal cavity outside the peritoneum within the mesenchyme and settling into the mesenchyme that is forming the mesonephric system. So we see that the gonadal ridge, or the primordium of the gonad, is formed by some of the resident mesenchymal cells in that area, and they are the receivers of these primordial germ cells, and that gonadal ridge does not extend the full length of the mesonephros. Here we see in cross-section a sketch of a 5-week embryo, and it again is illustrating the migration of the primordial cells from the yolk sac into the embryo, as we saw in panel A. And then here in panel B is a 3-dimensional sketch of the caudal region of this area, again the 5-week embryo, and we see the location and the extent of the gonadal ridges. So here is the mesonephros on the ventral medial surface. We see the gonadal ridge here, a mesenchymal ridge, and it has an irregular, deeper border that will be important, and we see the primordial germ cells kind of wandering into this area. Next, we'll take a look at transverse section showing the primordium of the suprarenal or the adrenal glands, the gonadal or genital ridges, and then the migration of the primordial germ cells into the developing gonads. So we said that we'll see more of the development of the adrenal glands as we proceed. And then finally, we'll see a transverse section of a 6-week embryo showing the primary sex cords So there's going to be changes in this irregular margin. And then finally in panel E, we'll see a similar section at a later stage showing the indifferent gonad and the paramesonephric ducts. So the stage is being set. So the yolk sac and the yolk stalk, of course, are related to the midgut, and we are having this migration of the primordial germ cells outside of the peritoneum and along the gut tube, and then up the mesentery dorsally, and then going laterally, and then finally climbing ventrally to get into position on this ventromedial aspect of the mesonephric region, to contribute to the formation of the gonadal ridge. The gonadal ridge mesenchyme is already there, and it's beginning to make up its mind to go ahead and do something. So we see structures we recognize then, the mesonephric duct that was developing in this area, and it will receive the vascular supply to differentiate into the complete system that we described before. It is functional, as we recall. There's another duct alongside that is developed, the paramesonephric duct. So the mesonephric duct and the paramesonephric duct are going to be important structures to keep in mind. And then we see the onlaga, if you will, or the primordium of the medulla, the adrenal gland, the suprarenal gland. And we see the sympathetic ganglion forming over here. And migrating away from that are other neural crest cells that will join the mesenchyme, forming the cortex of the adrenal gland. And eventually this blue structure is going to surround the pink one. Pink will become the medulla, and the blue will become the cortex. So the adrenal gland is forming right in this area. As we know, it's going to be the trigger for the termination of the dorsal or caudad migration of the metanephros. It is also associated with the general area of the gonadal ridges. They extend caudal and, cephalad, rostral, and caudad or caudal to this site of the development of the adrenal gland. So this is just a migration. I am not familiar with all of the literature as to exactly how much we know about these cells receiving signals and basically making the determination and gaining the knowledge, knowing when they should move, what route they should follow, and where they should end up. It's really quite an amazing thing, but I think reproduction is quite an amazing thing, so we shouldn't fall off our chairs just yet. We should wait and see more of this marvelous developmental system as we proceed. We are now, again, looking at this, and we're beginning to see that irregular border of the mesenchymal contribution to the gonadal ridge. And it's starting to have deeper indentations into it. Actually, these protrusions are elongating, and they're beginning to form the primary sex cords in the cortex. So this is the cortex of the gonadal ridge, and obviously we're seeing the medulla of the testes or ovary. So there's the surface and the deep portions. And then ultimately we see that these become even further elongated, and still are the cords, and they become associated with the primordial germ cells as they reach out to get in touch with them. So it's at this point, too, that the gonadal ridge is beginning to become more self-contained or pinching off. So we see there's even a urogenital mesentery. Again, a mesentery simply has to be a structure that will carry neurovascular bundle, and certainly neurovascular bundle is coming into here. We know that, even though it isn't illustrated. So here is figure 1329 from the textbook. I'll just skim through the legend and then take you through the illustration. So it is obviously going to be showing us the gonadal ridge. Excuse me. And so this is a photomicrograph of an actual stained histological sample, probably a 4 to 6 micrometer thick paraffin or wax embedded section that was cut with a knife and mounted on a slide and stained. It's through the abdomen of the embryo, Carnegie stage 16, about 40 days. So we're close to the time that we were just discussing in the latter panels of the illustration, prior illustration. And we see the gonadal or the genital ridge right on the surface. So the mesenchymal cells are condensed in this area and a little less condensed, a little bit deeper in, but we still have mesenchyme in this area. Peritoneal cavity would be located here. And then this is the dorsal mesentery of the gut, hindgut over on the left. Excuse me. So the most of the developing gonad is composed of the mesenchyme from the coelomic epithelium of the gonadal ridge, and the large round cells in the gonad are the primordial germ cells. So here's one that's quite distinctive that we can make out, and others that have a very prominent large nucleus that we might feel comfortable with that identification. As we proceed deeper into this section, moving dorsally, we see the mesonephros back here, and this is the developing glomerulus of the mesonephros. We don't have the mesonephric duct or the paramesonephric duct present in this view. It may be, they may be present, but farther dorsally. We can make out a cross-section through part of the tubule that might even be associated with this glomerulus that is formed. So the mesonephric tubule is seen in part here, and these other images are parts of mesonephric tubules and mesenchyme as well. That's just one where we can pick out the radial arrangement of the nuclei and the cytoplasm with the center where the lumen of this tubule would be. So let's come up with a definition of the terminal genital ridges and describe their formation in the embryo. So they're a pair of longitudinal ridges, as we said, running cephod to caudad, or rostrally to caudally, not the full extent of the mesonephros, but on the medial, and I've been adding that ventral or anterior medial aspect of the mesonephric ridges. They're formed by proliferation of the coelomic epithelium and a condensation of the underlying mesenchyme. So there's a combination of contributions here. These are all of mesodermal origin. The coelomic epithelium is of a mesothelium, and it is of mesodermal origin. But just it went in one direction, and now it's going in another direction to meet up with stuff that went in a different direction originally, but is going to join together to go in a final different direction of differentiation now that it's talking to these guys that came back together. So kind of like catching up with your old college roommates and starting a law practice together or a medical practice together, that type of thing. So who knew exactly what your lineage was way back then? You had some idea, had to go in different directions. When you finally meet up again, you can do make one concerted effort to form something new. So this is not an uncommon event. The development of the eye and the orbit are rather similar, and we might have an opportunity to talk about that later. So where do the primordial germ cells first appear? So along with which route do they migrate into the gonad? So again, for review, they first appear among the endodermal cells in the wall of the yolk sac close to the allantois, and from there they migrate along the dorsal mesentery of the hindgut and invade the genital ridges in about the 6th week of development. So in our mind, we're envisioning, again, week 2, 2 cell layers thick, week 3, an embryo, 3 cell layers thick. By the end of week 3, we're pretty much on our way to the organogenesis part of the digestive system and the other structures that we talked about. Week 4, 5, and 6, man, there's a lot of things happening with nervous system development that we haven't focused on yet, gut system development that we have, respiratory system, the facial development, all those types of things. And so there's a tremendous amount of cellular activity going on during this time. And so it is an era of significant and very active organogenesis. So it makes sense that by the 6th week, we're going to have the primordial germ cells where they're supposed to be, because after this, organs are going to become encapsulated in mesenchymal connective tissue structures, and they're going to be put together, have their hilus with a neurovascular bundle, any tubular connection to whatever else they need to connect to if they need to, and then they're pretty much on their way to final establishment and refinement and so on and so forth through the fetal stage. So by 8 week, if we zip right down to the bottom of this slide, we see that we've got commitment along the male lineage, and the interstitial cells of Leydig are beginning to produce testosterone. So by fetal stage, we got to have everything in place for that ultimate refinement of organogenesis along one lineage or another, specifically here considering the gonads. So they're first identified, the development of the testes, they are first identified with the presence of the dense fibrous connective tissue capsule, the tunica albuginea. So this is what I said. The mesenchymal connective tissue capsule is going to begin forming, and it's quite thick on the gonads, and so things have got to be in place before that happens, or stuff that might need to migrate in just might not get in. Seminiferous tubules develop in the sex cords, and then they remain inactive until puberty. We know that, and this is, again, pointing out the obvious. And then testosterone will begin to be present in the 8th week, and it has been stimulated by the human chorionic gonadotropin, and will continue to be stimulated by that throughout weeks 8 to 12. So this is a very busy slide, and I believe I have pulled out each of the subsequent panels to take a look at. So we will just quickly review it here, and then advance through some of the other slides that show us some of these events. So this is telling us about the development of the testes and the development of the ovaries, so different lineages altogether. So let's do the development of the ovary first. And again, we see the neural tube, so we know it's dorsal. There's an aggregation of neural crest cells about ready to develop into the spinal sensory ganglia. And we've already had some migrate out to form the sympathetic ganglia, and the primordium of the adrenal medulla, and the primordium of the suprarenal or the adrenal cortex is present here already. So we recall this image. We've already had the earliest migration into this area, actually completion, basically. So we're recognizing the mesonephric duct and the paramesonephric duct, and the genital or gonadal ridge, and the primordial germ cells located within these indifferent gonads. If there is no TDF, testes determining factor, then the development will follow the lineage for the female. So by week 12, so week 5, and then we're jumping to week 12, so quite a bit of time has gone by here. See, the adrenal cortex and medulla haven't quite finished what they're supposed to do yet, but they are pretty much left behind as we have the encapsulation of the gonad and its connection a mesenteric connection because it will have its neurovascular bundle coming into its hilus. See that what came along with it is the paramesonephric duct, and the mesonephric duct and the former primary sex cords are shown here. So just quickly we can go over to the male, and we see the sex cords that had developed and penetrated deeply and engulfed the primordial germ cells. And we pretty much in our mind know that these are going to end up producing the seminiferous tubules, and we see at this time they're called seminiferous cords. So they are obviously not going to persist in the female. We know that the mesonephric duct contributes to the male duct system, so we're going to expect that not to persist in the female, and the paramesonephric will apparently contribute something to the female reproductive system. Otherwise, why would we have it on the slide? So we're using an intuitive approach to these events. So the cortex of the gonadal ridge is seen here in the lighter pink. I just quickly checked in the male to see if they still use the pink, but they're not using color to differentiate genders here. This is just the mesenchymal surface cells that had mixed with the penetrating epithelial cells, as we had stated, to form the cortical cords now, and they become associated with the primordial germ cells. And this is the lineage that will, this is the cytogenesis and morphogenesis and organogenesis that will happen over these 7 weeks if there is no testicular determining factor. Obviously, if there is, we see that the thick connective tissue forms on the surface, the tunica albuginea, excuse me, and the cords are now the seminiferous cords, and they're beginning to contribute to this tubular connected system that we are recognizing somewhat as the adult. So this slide is now showing us what happens in the further development in the male. The seminiferous tubules will finally develop. The tunica albuginea mesenchyme will push deeper in to form septa, and these will surround these seminiferous tubules. So there are many of these seminiferous tubules. Remember, we're just looking at a two-dimensional plane of these. And then the reedy testes is going to develop, where the mesonephric duct has made contact with some of these cords. And there will be the persistent connection between the reedy testes and the remnant of the main mesonephric duct, what will now become the duct of epididymis, or the epididymis, and then these are the ductili efferenti that come in there. So we're recognizing a pattern that is very similar to the adult male gonad. Inside the seminiferous tubule were the captured spermatogonia, and I don't know why they have the interstitial cells of Leydig labeled inside there. I am going to use my intuition and say that I think that's an inappropriate label, because we have a connective tissue capsule forming around the seminiferous tubule, and the interstitial cells of Leydig are on the outside, in between the seminiferous tubules. So I'm just not going to believe that at this moment. The spermatogonia are definitely in there, and then the future sustentacular or Sertoli cells are in there, and they're, of course, going to form the epithelial lining of the seminiferous tubules. So what is called the indifferent gonad? In the embryo at the beginning of development, it is not possible to differentiate between the male and female. Hence, they are called as indifferent gonads. Primitive gonads consist of primitive sex cords and primordial germ cells. So they cannot recognize a difference. Even though we might say there's a cortex and a medulla, neither one has begun to develop to a state recognizable as one gender or the other. What is the first step of differentiation of the indifferent gonad in the male and in the female direction? So in the male, under the influence of the Y chromosome, those primary cords continue to proliferate and penetrate deep into the medulla to join the promesonephric duct, the mesonephric duct as we saw, and it will form the testes or medullary cords. In the female, in the absence of the Y chromosome and presence of double X chromosomes, the primitive sex cords are broken up into irregular clusters containing groups of primitive germ cells. So we know that these primordia of the oogonia are going to have to be free, not entombed, if you will, in a tubule, because eventually, as they develop as follicles, as we studied early in the semester, mature follicles are going to have to be able to migrate to the edge and break through the surrounding connective tissue capsule of the ovary at ovulation. So then the ovaries will develop. In week 10, they're first identifiable. Week 16, the primordial germ cells are found as the clusters of the primordial follicles with the oogonia inside, and of course they are active mitotically in the female fetus. So the primary germ cells have moved into this area. If they're going to differentiate along the lines to become oogonia, there's still not enough of them, so the oogonia have to do their job and undergo mitosis. And we know that that oogonia don't exist in the adult. So there's going to be a point where the oogonia complete their mitotic division and they are no longer present, and all we have then are primary oocytes. So here's the lineage from that illustration toward the direction of the female. The reedy ovary, the mesonephric duct and tubule are degenerating. They will be removed and eliminated, and the cortex is sending these cords in deeper, the cortical cords, and they're going to become associated with the primary ovarian follicles, ovarian follicles. And if we look close at this, we see the oogonia, and of course they are going to be mitotically active in and amongst the stromal or connective tissue cells, as we have described before. So here is the histological sample that we can look at and make determinations about what is what. So here on the left, we see some, predicting that these are even glomeruli in the mesonephros and the mesentery of the gonad, and there seems to be these cords present in there that radiate toward the center. And then here on the right panel, we see something is persisting in development back here, and this might be some type of a renal structure there. We see a mesentery attaching to it, and this structure that has this type of an appearance. So it doesn't seem to have well-organized, significant and recognizable cords pushing down in, but if we go to the cortex, we can kind of see some cords pushing in, but not as distinctive as on the left. This is already telling us that left is male and right is female. When we go to the higher magnification, what we can see are these clusters of very large cells with stromal cells dispersed around them, and then a relatively thin layer of connective tissue on the surface. It actually would be covered, as we know, with visceral peritoneum. And These obviously are ova. Here we can see the final division, mitotic division stages of these oogonia here. So there's going to be a rapid division of these oogonia by this point. Then on the left, we can see a lot of stromal connective tissue cells that go in. It's rather thick out here and go in and infiltrate themselves in between these structures that seem to be organized. A nice cross-section there, circular, with the hole down the middle. So it's an obvious tubule. And others nearby as well. This one cut as it bends around. So we see the lumen here and the lumen here. And this one we see a near cross-section. And within the walls, we see these larger cells and some smaller cells. Same here, same here. So these are the, we'll call them spermatogonia within the seminiferous cords. Higher magnification of this sample up here. So even from looking at illustrations, if we see an actual histological sample, we are capable of distinguishing the male from the female gonad at that stage. So we would predict that somewhere out 10 weeks or farther, because we just saw that the ovary is not really distinguishable until 12 weeks. This illustration is showing us the number of eggs in the human ovary at different stages of development. And we are looking at months after conception, so 3, 6, 9, first trimester, second, and third. Birth obviously occurring here, and then we change our scale to years, 10 years, 30 years, and 50 years. The x-axis is on scale of 0.5 to 7, number of germ cells times 1,000,000. So 7 million would be present, and it says in the human ovary. So we are going to interpret that as a single ovary at 6 months of development, so well into the fetal stage, obviously. And this high number is due to proliferation of the oogonia. The mitosis will begin to terminate at this time, and then there will be this spontaneous cell death called apoptosis, or programmed cell death, and millions of these will disappear up to birth, where at birth there is somewhere around 2 million present in the ovary. And then by puberty, somewhere here past 10 years, there isn't quite 500,000 of the primary, calling it number of eggs. We know it would be primary oocytes at this time. So think of the poor graduate students or undergraduate students that had to count all these eggs inside ovaries from sample tissues. Well, they didn't have to count that many. There are methods of estimating it. So this is most likely a reliable estimate based on morphometric analysis. And then by 30, this is when my wife and I started thinking about having a family. There was still more than enough left. You know, what's that? A couple 100,000. I didn't plan on having that many kids, so there were enough left over. And who am I to speak about my wife's ovaries and eggs? And she's now out here somewhere with me, and who knows how many she has left, but we don't intend to use them. We pretty much agree on that one anyway. So we recognize that there is a critical point out here at which, and actually it even is earlier, a point at which fertility becomes more difficult or compromised over time. We also recognize from this that this is the number of eggs, so primary oocytes or otherwise, not oogonia. So there is a vast number at birth, and those that you have with you from birth that persist to the age of 50 are as old as you are, basically. So this is if this is one egg left in the ovary at 50, that egg is also 50 years old. So it has not been a cell that has been renewed. The cytoplasm is old in it and all of that. So we know that there is aging of ova in females. So there isn't mitosis, so carcinogens can't affect, or whatever toxins and all that kind of stuff can't get to the DNA when there is tremendous mitosis of the oogonia, unless, of course, it occurs back here. Whereas in males, we remember that the spermatogonia undergo mitosis throughout life, so that can be impacted. So every mitotic event, DNA becomes susceptible, is the way to think about it. So there is the aging of the non-mitotic cells in female and aging of the mitotic cells in males. So from which components does the suprarenal gland develop, and where is its primordium located? So the medulla is the ectoderm, the cortex is the mesoderm, as we had pointed out. Ectoderm giving rise to the neural crest. The cortex, the coelomic epithelial cells located between the root of the mesentery and the developing gonad, begin to proliferate and penetrate the underlying mesenchyme, giving rise to the primitive cortex. So we've seen this all along. The medulla, parallel to the development of the cortex, cells originating from the neural crest cells invade the medial part of the cortex, giving rise to the medulla. So eventually, the cortex has to squash around the medulla. From which structures does the female genital tract develop? So obviously, this is the paramesonephric or Mullerian duct and the urogenital sinus. So this slide, this statement is included here to remind us that we are advancing to that discussion. And we saw the paramesonephric duct. Now we introduce this term, Mullerian duct, as part of that. So here we see an illustration. On the top left is the actual size, and it is only useful as being a reference to actual size when we look at this here. You may be looking at it as a little 1 square inch or 2 square inch on your monitor and so forth, so we keep that in mind. But this is stage 23, which is equivalent to 27 to 31 millimeters in length. 57 days, and we recognize this as we had talked about the urinary system. So we see the lobules of the metanephros, the metanephric duct, which is now the ureter, is coming into this cloacal area. Cloaca has been separated into the urogenital sinus, with the bladder forming in this area. And obviously the urethra is going to begin to form down here as we see the paramesonephric duct in blue and the mesonephric duct in yellow joining that. So we know it's going to be urogenital area emerging from this ultimately. The genital tubercle is up in this area. So that's that ventral part of the cloaca that was separated from the dorsal part by the urorectal septum that we mentioned, and the rectum developing back here. The anal membrane is present, and we know that there is a membrane covering this area as well that does persist in the female, and that will be part of our discussion in the future. So I like this illustration because it shows very nicely the metanephros has now migrated up to the rostral pole of the gonad, the gonadal ridge. It's not a ridge anymore, it is the gonad. And we see the paramesonephric duct and the mesonephric ducts right nearby, so it's still a bit indifferent from this initial visualization of it. And We know that the adrenal gland was developing somewhere near the rostral pole of the gonad. So this metanephric structure, the final kidney, has met with that somewhere here, and it should have terminated its migration. We see a little farther inferiorly here, again, the blue duct, the right and the left paramesonephric ducts are fused in the midline. So they will persist as the female, and they will contribute to the formation of the uterus and the fallopian tubes. The fallopian tube would open to the female gonad as we recognize. So everything's in place. We're just waiting for the chemical signals. Now we proceed to 8 weeks, so the early fetal period in this view. And it's 35 millimeters long. Again, they give, for sake of reference, the upper panel here. We can see the umbilicus, the umbilical cord there. So the paramesonephric duct is now differentiated along the lineage of the female, very obviously. And the uterine tube has formed, and the uterus has formed here. So obviously, some of the urogenital sinus is going to contribute to the formation of the vagina. For this to be female, we have to separate the urethra from the vagina. The remnant of the mesonephric duct is degenerating, continuing to degenerate, as is the mesonephros nearby. And the metanephric kidney persists, and the ureter coming down to drain in to the bladder. So we see the structures are quite well developed, or established, let's say, and the genital tubercle is now recognized as the clitoris. Here at 3 months, we now see, again, the kidney. It's lobulated. Metanephric, ureter, into the bladder. The bladder has now been separated from the part of the urogenital sinus that developed into the vagina, and the urethra is forming anterior to the vagina, and of course is open into the bladder and continuous into the ureter. So the trigone is will have formed somewhere here, the opening of the urethra, and the two. We see the left one here, and we know the right one's on the other side. The two ureters is the trigone, 3 corners of the triangle, the trigone. The bladder is here, and the urachus is the term for this structure that now the former allantoic stalk is called the urachus. Continuous with the bladder, that obviously becomes obliterated at some point because we don't pee through our belly button. As I stated very early in the semester, sometimes this does remain. Can become patent again in pregnant females, and droplets of urine will present at the umbilicus. The rectum is well formed at this point, and we see the clitoris has formed more completely. And then here we see at term, so the former cloaca now has gone from an anterior presentation, that tail curve, to a more vertical positioning with an antero-inferior and directly inferior presentation. We now see the ovary at the pelvic brim here, and we see the fallopian tube, the uterus, the cervix pushing into the upper vagina, and the vagina, of course, having the hymen covering it, so that persistent portion of the cloacal membrane, and the labia minora and labia majora have fold, as well as the clitoris, with the urethra anterior to the vagina. So we end our lecture on that slide, and we will continue our discussion. My PIP is going crazy. I'm still here. And we'll continue with the next lecture to discuss the development of the genitalia, and that will be pre-recorded and posted. Appreciate your time and attention. I'm going to check out since there won't be any student questions. So I'll send Nayeli an email now, and we'll get that initiated, okay?
Sounds good, sir. I normally stick around for questions, but unless you've got something, I'm going to go ahead and head home since there's nobody here.
Sounds good to me.
Have a good one, sir.
And me down there, and me over there, and me up there.
Yeah, all of you guys.
Bye.